Hello, hi everyone. I'm Sandhya. I'm your botany lecturer. Now in this video, in this lecture, we are going to study about the endomembrane system. The membranous cell organelles which are interconnected and having the distinct structures, and function, and the functions are coordinated. So all the cell organelles are together forming as endomembrane system. Endo means inside, membrane, they are all membranous cell organelles. So in the endomembrane systems, they have distinct structure and function, and the functions are coordinated. Okay. So here the first one is endoplasmic reticulum. The endoplasmic reticulum is also called as ER. Then Golgi complex or Golgi body or Golgi apparatus, lysosomes and vacuoles. So these four form the endomembrane system. Whereas the mitochondria, plastids, their functions are not coordinated. They are having different functions. So hence this is the endomembrane systems. First we will deal with the endoplasmic reticulum. This endoplasmic reticulum ER was first coined by K.R. Potter. K.R. Potter. Okay. We are studying about endoplasmic reticulum. The term endoplasmic reticulum was coined by K.R. Potter. K.R. Potter. They are thread-like structures which are present inside the cytoplasm. So if you take the cell, if this is a cell, this is the cell, this is the nucleus. So near the nucleus, the endoplasmic reticulum is going to be there like this. Like this, it is going to be present. Like this. Okay. They are thread-like structures. They are tubular structures which are just scattered in the cytoplasm. Then they divide the intracellular space into two distinct compartments: the luminal vesicles, and the extraluminal vesicles. This is the extraluminal vesicles, the cytoplasmic vessel, and this is the luminal vesicles. Okay. Luminal vesicles inside the endoplasmic reticulum. The extraluminal vesicles means the cytoplasm one. The cytoplasm one compartments, two compartments, the thread-like structures which are just scattered in the cytoplasm and nearer to the nucleus. They are the extensions of the nuclear membrane. They form like a network-like structures inside the cytoplasm. That's why network. No, that's why reticulum. Reticulum means network. Reticulum is network. Then the endoplasmic reticulum is of two types. The endoplasmic reticulum is of two types: a rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum means here on the surface the ribosomes are present. On the surface the ribosomes are present. The ribosomes are attached to the ER. So hence it is called as rough endoplasmic reticulum, which are having the ribosomes. Hence they play an important role in the protein synthesis, because ribosomes are important for protein synthesis. As this particular ER is having ribosomes, it is called as rough ER. It is helpful in the synthesis of proteins. And smooth ER is not having any ribosomes. Smooth ER is not having any ribosomes, but it is very much important for the synthesis of lipids. This is about the endoplasmic reticulum. Coming on to the Golgi body, Golgi apparatus, or Golgi complex. These Golgi complex was first discovered by Camillo Golgi. Camillo Golgi. So in order to honor him, in order to appreciate him, these particular structures are called as Golgi complex. Golgi complex. They are flat, disc-shaped cisternae. They are flat, disc-shaped cisternae are present. They are present somewhat nearer to the nucleus, So if you take the structure of a cell, this is the nucleus, this is the reticulum, endoplasmic reticulum. So here somewhat nearer to the nucleus, Like this they are going to be present. This is the Golgi complex. Now this is the Golgi complex. Okay, they are stacked parallel to each other. They are stacked parallel to each other. They are stacked parallel to each other like this. Okay, they are flat, disc-shaped or cisternae Then these cisternae are concentrically arranged near the nucleus with distinct convex cis phase and trans phase. Distinct convex cis phase forming phase, and the trans phase, which is the maturing phase. Cis phase is the forming phase, the fresh one. Then trans phase, the trans phase is the maturing phase. Okay, then this particular Golgi apparatus performs the function of packaging materials to deliver either to the intracellular targets or secreted outside the cell. Okay, outside the cell. They are in close association with the endoplasmic reticulum. They are very much in close association with the endoplasmic reticulum. Okay. Here also the endoplasmic reticulum will be there. So they are in close association with endoplasmic reticulum. That's why it comes under endomembrane system. For the functions are coordinated. How they are coordinated? A number of proteins will be synthesized by the ribosomes, which are present on the rough ER. Okay, rough ER, and these are modified into cisternae of the Golgi complex. These are modified cisternae of the Golgi complex. This Golgi apparatus is useful for the formation of glycolipids, glycoproteins, and glycolipids, for the synthesis of glycolipids and glycoproteins. And also it is useful for the formation of the cell wall materials. It is useful for the formation of cell wall materials. And also it is useful for the formation of cell plate during cell division. This cell plate later on is going to form into middle lamellum. Middle lamellum is going to transform into the cell wall. So it plays an important role in the formation of glycoproteins, glycolipids. It also synthesizes the cell wall materials, and also it helps in the formation of cell plate during the cell division. This is about the functions of Golgi apparatus. Coming on to the lysosomes. Lysosomes are the structures, are the membrane bound structures only. They are membrane bound structures. Lysosomes. Now we are studying about lysosomes. They are membrane bound. Yes. Membrane bound vesicular structures. Structures which are formed by the process of the packaging in the Golgi complex. Okay. The cell, the nucleus. This is the endoplasmic reticulum. Like this. Here the Golgi complex will be there, and here the lysosomes will be present. Okay, lysosomes will be present. They are in coordination with the functions are in coordination with the functions are in coordination anamata. Okay, so and first nearer to the nucleus, endoplasmic reticulum, rough ER, then smooth ER, then a Golgi complex will be there. Then by the formation of the packaging materials of the Golgi apparatus, the lysosomes are formed. Then these lysosomes are going to have hydrolytic enzymes, which are going to digest the carbohydrates, proteins, and lipids. They are having hydrolytic enzymes or hydrolases, which are going to digest the carbohydrates, proteins, lipids, and nucleic acids also. So hence these are called as hydrolases enzymes are present. Then the enzymes which digest the carbohydrates are called as carbohydrases. The enzymes which are going to digest the proteins are called as proteases. The enzymes which digest the lipids are called as lipases. The enzymes which digest the nucleic acids are called as nucleases. Okay, nucleases. So what they're having? They're having hydrolases. These are the hydrolases, or which can say hydrolytic enzymes, digestive enzymes, like this. Then they are having a acidic pH. These lysosomes are having acidic pH. Then under extreme conditions, under starvation conditions, what happens? These lysosomes are going to release the hydrolytic enzymes, and that is going to digest the cellular contents, causing the death of the cells. So it is going to cause autolysis or autodigestion. So during digestion, what happens? For the digestion of the cellular contents, it is going to release the hydrolytic enzymes. Or suppose if a cell organelle has become old, or that particular organelle is not working properly. So what happens? That organelle comes near the lysosome, and this lysosome is going to take up and is going to digest that particular thing, or digest that particular cell organelle. Or when starvation conditions are there, this lysosome is going to release the hydrolytic enzymes and causes the digestion of the cellular contents, and ultimately causes the cell death, which is nothing but the autodigestion or autolysis. And hence these lysosomes are also called as suicidal bags. Because it's going to cause the autodigestion. Hence they are called as suicidal bags. These are nothing but the lysosomes. Coming on to vacuoles. Vacuoles are also membrane bound. Coming on to vacuoles. Vacuoles are also membrane bound spaces which are found in the cytoplasm. In plants, there are bigger vacuoles are present compared to animal cells, and these vacuoles are going to have water, minerals, excretory materials, secretory materials, waste materials will be present. In some plant cells, they also have some pigments also. So these vacuoles are going to have water, waste materials, then excretory materials, excretory products, metabolic byproducts are present. In some plants, pigments are also present in the vacuoles. Those pigments are called as anthocyanins. They impart color to the plant part. They are single membrane. The vacuole with a single membrane is called as tonoplast. This is a neat question. Once it was asked. The vacuole with a single membrane is called as tonoplast. And the plant cells occupy 90% of the vacuoles. The vacuoles occupy 90% of the volume of the cell, and they play an important role in osmoregulation. Osmoregulation means the content of the water. They regulate the water content. In plant cells, the tonoplast is going to facilitate the transport of the number of ions and other materials against the concentration gradient. Okay, so that is about the vacuoles, which is very much important for the osmoregulation. And in amoeba, contractile vacuole is present. Contractile vacuole is going to be present, which performs the function of excretion. Contractile vacuole. Then in protists, food vacuoles are present. Where they are going to engulf the food. These particular food vacuoles are going to engulf the food. Okay. In amoeba, contractile vacuoles are present. This is about the vacuoles. Thank you. Now just outline of this lecture. So endomembrane system includes endoplasmic reticulum, Golgi apparatus, lysosomes, and vacuoles. They are they are membrane bound, and they're closely related in terms of structure. At the same time, their functions are coordinated. Hence, these are included in the endomembrane system. Whereas the mitochondria and chloroplasts are having different functions, so they're not included in endomembrane system. So this endoplasmic reticulum is related with the protein synthesis. Then this endoplasmic reticulum will be in connection with the Golgi apparatus. This Golgi apparatus is useful for synthesis of glycoproteins and glycolipids. Then this Golgi apparatus will be in association, somewhat in association with the lysosomes. Okay. By the process of packaging in the Golgi apparatus, these petal lysosomes are formed. These lysosomes are nothing but the suicidal bags of the cell. They are helpful in autolysis and autodization. So they are suicidal bags. Then vacuoles. Vacuoles are also membrane bound. They are having water, waste materials, excretory materials, metabolic byproducts, byproducts. Sometimes in plant cells, in some plants, the vacuoles are having anthocyanins, pigments, and it is membrane bound, called as tonoplast. In amoeba, contractile vacuole, which is useful for cell excretion. Food vacuoles are also present, which are useful for engulfing the food materials.